Okay. Hi, everyone. My name is Dean. I'm the primary developer behind OpenIMC and a graduate student at USC. And today I'm going to be doing a quick walkthrough on how to install OpenIMC, some basic features, and a general tutorial about how to potentially analyze a data set end to end. So to get started, I'm just going to share my whole screen. So here I just have Safari open and I'm going to go to GitHub and you guys will also navigate to the OpenIMC GitHub. And so in here, what you'll see is all of the source code for GitHub or for OpenIMC. It is open source. You guys are free to fork it, make any changes that you desire, and if any of them you think are beneficial to the community, contribute to the open release. To install OpenIMC, we have a very quick installation guide here, which relies on the UV package manager. If you're more used to using Conda, we have instructions inside of our documentation available here. You can simply copy this in order to set up OpenIMC and then just navigate into your terminal. Here, I had previously installed it and just cleared it. And so now I will reinstall it just for you guys to see. So I can simply paste all of this into the terminal and run. And if I do this, you will first see that I am cloning into the Git repository, then UV is getting set up and it's now setting up our environment. So we created a virtual environment at -ben. We then activated the virtual environment and we're now installing all of the various packages that OpenIMC relies on. And one great thing about using UV is how fast it is. You can see how quickly we were able to install all of these packages that we need. And boom, we're done. Conda would have taken much longer, and that's why we recommend it. And then the last thing is that we actually built the OpenIMC package. So once you've built the OpenIMC package, the next thing to do is to just run OpenIMC-GUI. And this is how you run OpenIMC. You can also run it in a number of different ways, but this is the simplest way in order to get into the user interface, and that's what GUI stands for. So if I hit enter, it's going to take maybe 10 or 30 seconds to populate the screen. This is really just a bunch of backend stuff that's happening so that when you go to use OpenIMC, everything works as expected. And so we're just going to hang out for 5-10 more seconds, and then OpenIMC will open for us to use. And I also encourage you all to go to our documentation over here and read it through thoroughly, which is going to be much more thorough than anything that I'm able to show in the span of a single video. You can see that OpenIMC is opening, and we do think that that is slowest on Windows, second slowest on Mac, and then by far the quickest on Ubuntu. So here we have OpenIMC. Mine is in dark mode because my computer is in dark mode, but if you have this in white mode, this would be white. So the next thing to do is to open a file that we actually want to analyze. So here you can import data in two different ways. If you're familiar with IMC data analysis, a lot of downstream tools rely on an OMTIF export of raw IMC data, which can be generated with various tools, including MCD Viewer, or you could use the raw IMC data through the .mcd file, and that's what I'm going to do. So I'm going to say I want to open a .mcd file, then it will open my file explorer, and I'm just going to navigate to a .mcd file that I have on my desktop, and I'm going to click enter to open it. Now we're going to read in the IMC data, and here we have the first ROI in our IMC data analysis, The DNA1 channel will auto-populate. You can see if we scroll down, it's selected. Here are the channel selections for the main viewer. So the viewer, for instance, I want to add CD45, boom, I can do this. If I want to view the channels in grayscale, I can do that if that's preferable. And then if you want to save this, you can just hit save this and it'll save the canvas like so. If you want to zoom in, you can do like this. You'll zoom in both fields of view, and then you can just click home to go back. You can also look at all of the channels in the acquisition at once. You can see that my ROIs are actually quite small. And so I can actually just make them bigger by hitting up on these arrows. And now I have maybe a bunch of ROIs that I can actually see. And then I can scroll through these and look at all of the channels at once. And I can also scale these by just applying a quick contrast scaling. And now I have a bunch of markers that I'm looking at all at the same time. Cool. So that's the crux of the main viewer. Some other useful things are scale bars. You can change their length very easily. Here, where your maximum is set to be some 30% of the actual image size. So I couldn't go to 50, but I can go to 20, for instance, 1 or 10. You can also denoise the image. So if you have some hot pixels or something, you can hit, for instance, I want to remove hot pixels in my CD45. Or if I want to do it to both channels, I can hit apply to all channels. And if I want to turn off, I can just click this and then turn it off on all the channels at once. Or I can just deselect the denoise. You can also do a custom scaling. So if I wanted to do, for instance, in CD45, that the maximum is 30, so that's 3, and then I add 0, 30, and it'll update in real time. And you can also just reset back to the main. So that's very nice for scaling your images so that you can view them. And then if you wanted to create a composite image, you could first turn off grayscale and then turn off the grid view. And so the next thing would be to, for instance, say, okay, I have my DNA channel and I want that to be blue. And now I have my CD45 channel and I want that to be green. And now I have a very nice composite that I can just hit save on. But I can also, in my custom scaling, like we just saw, I could say, okay, I actually have my green channel. The maximum is 64. I actually want that to be 30. And in my blue channel, my maximum is 200. I actually want that to be 50. And then you can control what these images look like. And then just disable it like so. Go back to the grid view to turn off the RGB. You'll also see that here you have all the metadata associated with your acquisition and the types of performance system that you're on, and this is relevant for downstream steps. So for instance, segmentation would rely on having a GPU, or if you have Mac, like I'm using, an Apple Metal performance shader, which is Apple's version of NVIDIA's CUDA that you can then run deep learning models on. So let's go to that. So for segmentation, we offer numerous tools: deep cells, cell-SAM, cell poses different models, Cyto3 and nuclei, the ability to run an elastic model or to just use watershed. We strongly recommend using cell-SAM. It has generically very good performance on IMC data. It was trained with IMC data. And so then the next step really is to select the parameters for segmentation. So the first one is the segmentation channels. So here you have to select nuclear channels and cytoplasmic channels. You'll notice that the DNA channels are automatically selected for the nucleus. You can, of course, deselect them or add additional ones. And then cytoplasmic channels you need to select. And so for the purposes of this, I'm just going to do a quick example where I select CD45, vimentin, cytokeratin, and I'll also add 45RO and 45RA just to complete my sample. And then I've now selected my channels and I can choose how they're combined. So I can combine them if I go back into here by taking the mean, or I can do a weighted addition at the maximum value at each pixel or a PCA. You can also here then say, when I combine the channels, when I take the mean, how do I combine them? So one of the approaches is most common is to do a channel-wise min-max. So you scale each such that the minimum and maximum values are 0 and 1, and then the mean is appropriate to use. But you can also do other kinds of normalization or no normalization. You can also denoise the image. So like I was talking about in hot pixel removal earlier, you could apply the hot pixel removal prior to any kind of segmentation occurring. And then for CellSAM specifically, you must provide an API key, which you can get from their website, which there's a link here. It's a free API, but you just need a key to download the weights. And then we have the parameters for CellSAM. So we have WSI mode. If you have a whole slide image, which is really large, you can tile that whole slide image so that you can then segment each of those tiles and then piece them back together. You can also use a contrast enhancement, or you can use this very nice parameter called gauge cell size, which will run the model twice for segmentation, estimate the error, and then run again to get a nicer, more accurate mask. So I'll use that. Then you have a few parameters here. One of them is just that when we're done with the segmentation to show the overlay. A second one is to save the masks. So you can actually load segmentation masks into OpenIMC. And so because segmentation is such a time-consuming process, it's often nice to save the mask and then only do this one time. And then for any subsequent analysis, load the masks back. And then you can also say that you want to segment all the ROIs. In this case, I have 106 ROIs in my MCD file. And rather than having to do each of them one at a time. For now, I'm just going to segment the ROI that I'm on. And I'm not going to save the masks anywhere, just for this example. So then I hit run segmentation. This is going to, if we look in the terminal, start segmentation, and in here we have this little progress bar. And so we can see that CellSAM has begun to run. CellSAM is quite slow, and so I would say that, again, I would really place an emphasis on doing the segmentation one time. It's going to take quite a while, especially if you have a very large data set, but then you'll just be done and you can come back to the same masks again in subsequent analyses. You can also look at progress bars for CellSAM inside of the terminal. But here we are. We finished the segmentation. And one thing I just want to note is I didn't really optimize any parameters for the segmentation. I kind of just chose randomly, to be honest, while we were making this video. And I got beautiful masks that perfectly encapsulate my cells. So now that I've segmented my cells, the next step is to extract features out of them. So you want to extract features that describe things like the morphology and intensity of each of your channels. So here it's going to segment all the acquisitions that have a mask, which in this case is just one of them. Then I'm going to say where I want to save my features to. So here I'm just going to save them to my desktop just for convenience. And you can change the name of the file that gets saved. You can also do a same denoising process that we used for segmentation in feature extraction. So if you want to get rid of hot pixels before, for instance, measuring intensity, you can do that by saying, hey, I want to get rid of hot pixels in every channel. You can also do spillover correction if you've generated a spillover matrix, and you can actually do that within OpenIMC, but I won't go over that in this video. And then you can also scale your features with an arcsine H transform, which is very commonly used in IMC data analysis so that all the channels are more comparable to one another. The last couple of steps here are which features you want to extract. By default, we're going to extract all of these morphometric features and all of these intensity features. But for instance, let's say you wanted to extract just the mean intensity in each channel. You could deselect the rest of them and just select the mean. And similarly for morphology. And then here you can also exclude any channels that you don't want to measure. So for instance, these ICSK channels are channels that Standard Bio offers for membrane, not really useful in terms of a marker for downstream analysis, so I can exclude those. Then I just hit extract features. Here it's going to use your multi-threaded CPU in order to really accelerate the feature extraction process. And we're already done with the feature extraction for this ROI. And you can see that now we ungrade some of these future steps. So batch effect correction and cell clustering. If we had multiple .mcd files loaded and we wanted to correct for a batch effect between them, we could hit this batch effect step here. And then we could say, okay, we want to use the features that we've already loaded. And if we had additional metadata connected to each acquisition, you could, for instance, attach that here and correct on that metadata. And then you can select the method that you want to use. So Harmony or Combat to do batch effect correction. And then what covariate do you want to correct on? So for instance, source file, which is the MCD file itself. So one slide. Or you could do the acquisition ID, or you could do custom groups. So for instance, in custom groups, I could say, okay, I want to make a new group, which I'll call batch one. And then I want to add this acquisition to my batch one group. And then I would do this if I had many batches that I wanted to create, and then I could correct for that. And then you just click apply batch correction and you can save the corrected features as well. I only have one acquisition, so I won't do that. After you've corrected your features, you could then go into the clustering tab. And in the clustering tab, you can then actually get some meaningful outcomes from your IMC data. So the first step is to set up the clustering. And so here we have the various feature sets, for instance, corrected or uncorrected. And then you have various methods of clustering. So Leiden, Louvain, k-means, HDV scan, and hierarchical clustering. And if you change these, the different parameters that you need to provide will change. We'll stick with Leiden. You may also want to scale your features, for instance, with a z-score, because you're going to be comparing maybe morphometric features like area with intensity features, and those could be on vastly different scales. And then the parameters for the graph, which is specific to Leiden. And we'll just leave that as is. So then I can just go over here, hit run clustering, and I can select what features actually go into the clustering. I'll make this bigger just so everybody can see. And so I can say, okay, I actually want to do area and eccentricity as my only morphometric parameters, but I'll keep all of my intensity features. One thing you'll note is that we excluded the ICSK channels earlier in the intensity extraction, and you can see they don't appear here. We don't have features that describe the intensity in ICSK. You'll also see that here we're defaulting to just the mean expression within a cell, and that's set up here in this intensity filter, but you can use either all the intensity features or various other ones, but means are generally sufficient. You can also exclude any cells that are touching the edge of an acquisition, which can be useful in terms of not having cells that are cut in half. You can also control for maybe artifacts, say I want a minimum area of 10 square microns and a maximum area of 200 square microns, and that would be a normal range of cell sizes. And then percentile censoring would just exclude any outlying features, which is another commonly done thing. So then I click run and oh, I got an error. The reason that I got this error is because I actually only have 13 cells in my ROI, but I set my number of neighbors to 15. So in order to overcome this, I would just go back to my clustering setting, change this and neighbors to, for instance, 10, go back to my clustering. All the parameters I set up earlier are still saved. And then I would just hit run again, and then boom. And in this example, I got 2 clusters, which are potentially not very informative, but that's because I'm only using one acquisition with 13 cells. There's not very much information available. But you can see I get this very beautiful figure that I can just theoretically immediately put into a publication. If you want to configure the plot a little bit more, you could say, I want to change various things about it, like the font size or the feature labels. So you can see, for instance, here we also have the metal tag appended to our feature name. That might be a little bit ugly for a publication. So I could just say, hey, I only want this to say CD14, and then that change will be applied. And then if I go back to the clustering, you can see that now I have CD14 here instead of CD14 underscore the metal underscore mean. And that's just kind of a beauty thing. You can also do the same thing for phenotypes. So I could say, hey, my cluster 1, those are actually T cells, and my cluster 2, those are actually B cells. Again, just hypothetically. and then that will show up in the plot. There's also various other plots that you can make. For instance, instead of a heat map, you can make a cluster map, which represents more of a mean inside of each uh, cluster instead of just the individual cells, and also enables you to do dendrograms and things like that. You can also make a U map. So, for instance, here we have a U map settings that we set up, and we want the number of neighbors, for instance, to be 10, and then a seed for the U map, and then I can hit run. Okay, UMAP is going to take a few seconds to compute, and then I got a UMAP, which of course with this low number of cells is not particularly useful. But here I can also color the UMAP by various things. So I could say I actually want to color by CD31 expression in addition to the cluster or various things. You would also do t-SNE. You could do box plots of the expression. So for instance, if I wanted to select my fibronectin marker, for instance, I could see, okay, T cells have really no fibronectin whatsoever. B cells have a much larger range of fibronectin expression than the T cell labels that I had set up earlier. And you can also do statistical tests. And we see that here we have no pairwise comparisons that were significant between these two clusters. So yeah, that's the crux of the clustering-based analysis and the cell phenotyping. And then we could also go to a spatial analysis. So here you set up your graph. So again, here I have a very small number of cells. So I'm just going to set K as 5 for my nearest neighbors. I build the graph, and now I built the graph. And so now I can do various things. I can do a pairwise enrichment, distance distributions, a spatial visualization, or spatial communities. Here I'll just show the spatial visualization. So for instance, if I generate a spatial plot, you'll notice that actually what we have here is each point is exactly where it appears inside of the image, but it's labeled now by the cluster. Or if I don't want to label by the cluster, but for instance, I want to label by CD45 expression, I can do that. And so for tissue samples, this is really nice to be able to generate a spatial visualization. And again, you can just save the plot. And I didn't say this, but in the clustering, you can also do that as well. We also have an advanced spatial analysis, which is based on SquidPy. And so this gives you a little bit more features at the cost of complexity. It also can be quite slow to load. And so here now we got that to load up. And so again, we need to remake a graph because SquidPy uses an AND data backend instead of just a normal network X graph. But so here I can build this graph. Now we built the graph. And so then we can do some other functions here like neighborhood enrichment where we say, okay, we have such that it looks like T cells actually don't tend to be next to one another. They tend to be, for instance, next to B cells or something like this. But this is, in this example, of course, not very rigorous. But yeah, so that's the core functionality of OpenIMC. There's obviously many, many more things that you can do. For instance, you can do quality control analysis. You can do pixel level analysis, ignoring cell level. You can do high resolution IMC. You can do spillover matrices. There's a bunch of stuff that you can do, and I encourage you to check it all out and really read our documentation because I think that's the best way in order to get through the full suite of features that OpenIMC offers for analysis. But I hope that you can understand how you could take an MCD file or a set of MCD files, load them into OpenIMC, segment them, extract features, and do some cell phenotyping and spatial analysis and generate really meaningful figures and results that could potentially go just straight into a manuscript because of the way that OpenIMC is constructed. So yeah, I think that's really everything. Maybe one last thing to show you is that when you close OpenIMC, it will ask you if you want to save a text file of what it is that you did while you were in OpenIMC. And this is really important for reproducibility and allowing people to redo exactly what you did. So if we hit yes, what it will ask us is, where do you want to save this file? So here, I'm just going to save it, for instance, into my OpenIMC directory. hit save, and now we saved that file. And if we were to navigate into the OpenIMC directory like so, we now have this txt file. And so now if we open this, you can see we have a record of everything we did. So the first thing that we did was cell SAM. So we segmented the cells, and it tells us exactly the normalization methods that we used, what channels went into the nucleus, what channels went into cytoplasm, how we combined them, and what settings we used to denoise each channel. And then the settings that we used for cell SAM itself and what the output was. So again, very useful for reproducibility. Same thing for feature extraction and its subsequent denoising. And then clustering, the settings that we used, the algorithm that we used, class annotation. So we annotated the cells as T cells and B cells, and then the spatial analysis graphs that we put together. So yeah, I mean, very useful for reproducibility, and I hope that you guys can really get a lot of value out of OpenIMC. And feel free to submit any issues into the GitHub if you have questions, and I'd be more than happy to answer them. Thank you.